Hello, I'm Professor Paul Bingham, and this is Biochemistry 1. The purpose of this segment is to take the first important steps toward mastery of the concept of allostery. Let me emphasize before we begin that this segment follows very naturally from the hemoglobin and myoglobin segment, and you can't really follow the discussion here if you haven't recently watched the hemoglobin and myoglobin segment. So if you've not, go back and do that before you begin. So the larger theoretical issue here, the thing that we're concerned with, is to use hemoglobin and myoglobin, and particular hemoglobin, to look not only at the function of monomeric globular proteins as symbolized by our little cartoon here on the screen, but more importantly how when you put several different protomers, individual globular proteins together to form a multimeric protein, the kinds of new capabilities that allows you to do. This is making multimeric proteins is apparently a simple evolutionary route to taking monomeric proteins and giving them new extra capabilities, regulatory capabilities. Let me remind you of what we saw in the last segment, the myoglobin and hemoglobin segment, that hemoglobin works in the circulatory system to move into the lungs and take oxygen, and then to move out of the lungs back into the tissue and release it to where it's needed for active, ongoing, continuous metabolism. And in contrast to myoglobin, which simply grabs whatever oxygen is in the neighborhood, hemoglobin you'll recall from the earlier segment, has this remarkable capability of absorbing lots of oxygen in the lung and then releasing lots when it gets to the lower oxygen need environment in the tissues. And we saw the kinetic evidence that was true. We learned how to think about it quantitatively in the earlier segment. Now we need to look at the molecular machinery that makes that remarkable and physiologically important property possible. So let's talk about this sort of the fundamental mechanics of hemoglobin cooperativity, the fact that the binding or the release of one oxygen to the hemoglobin tetramer influences the binding or release of oxygen from the other three protomers in the tetramer. Cooperativity. Remember, first of all, that non-cooperative binding, like to oxygen to myoglobin, for example, produces a hyperbolic plot of the sort that's diagrammed here, and a sigmoidal and cooperative binding produces a sigmoidal binding curve, as illustrated here. So again, let me flash those for you. Myoglobin looks hyperbolic because it's simple binding. Hemoglobin is sigmoidal because it's cooperative binding. So cooperativity requires, at least in the cases that we understand, requires a multimeric structure. So let's look at the two things that together make this cooperativity possible. On the one hand, protomers talk to one another, so that when an event happens at one protomer, the other three protomers in the tetramer know about it. They talk to one another in one way or another, and today we're going to look at the molecular basis of that conversation, if you will, that exchange of information. And an intimately related other thing that's necessary is that the individual protomer must in some way change shape in response to, for example, binding of oxygen in the case of hemoglobin, in order to communicate to the other protomers. In other words, there's no radio waves or microwaves or Bluetooth between protomers. There's only the contacts that they make in the multimer, and therefore for one of them to talk to the other, it must change shape and tug on those others in a way that influences their behavior in turn. We know of no other way to do it, and the experimental evidence indicates that that's how it's done, not only in the hemoglobin case, but more generally in other multimeric proteins that we'll see later engage in this kind of more complex behavior. Okay, so let's begin by looking at how each individual protomer changes shape, the molecular details, biochemical details of that, and then we'll come back to how that shape change is communicated to the other protomers in the tetramer. So the first and simplest example of how an individual protomer changes shape in response to, in this case, oxygen binding, is the distortion of the heme ring. So we alluded in the hemoglobin and myoglobin segment to the fact that when the heme iron binds oxygen, there's an exotic type of bond that causes the iron to move toward a +3 oxidation state rather than the +2 oxidation state shown here, and that loss of an electron reduces the size of the heme group slightly but significantly. That's very important because the +2 iron is actually a little too big for that pocket in the heme group, and it sticks up and distorts, pushes down the surrounding heme group. Let me show you what that looks like. So this is now that heme group in a cartoon form embedded in its context in hemoglobin. And notice at the bottom that in the oxygen-free state, it's flexed slightly, a little bit like bird wings flapping. It's flexed slightly because the iron is a little too big. When oxygen comes in, partially pulls an electron away from that iron, reversibly changing its oxidation state in a subtle way. It then reduces in size, and the wings flap back like this. And the iron settles back down in this dimension, down into the heme ring. What does that do? That iron is hydrogen bonded to the histidine F8, as we saw in the previous myoglobin and hemoglobin segment. That's central to this protein structure. So what does that do? That grabs on that histidine and pulls it down in this particular orientation. It grabs, therefore, the F alpha helix and moves it significantly. And all the other helices in that monomer, that protomer, are going to change to one degree or another as a result of the F helix being pulled in this way. So this is an internal change to the protomer, but an internal change to the protomer that will change its shape and therefore its tug on its contacts with the other protomers in the multimer. All right? And this is just another diagram of the two things side by side. Again, it's the change in size of the iron atom upon binding oxygen that deflexes the heme group, pulling on the histidine residue, pulling on the F helix. The knee bone is connected to the thigh bone, and so on. Okay, very straightforward, kind of a mechanical thing, but that's a signal that's being distributed through the particular protomer's structure, and therefore, by tugging on its contacts with other protomers, changing their behavior. Much more about that momentarily. Let's look at a second set of changes that are relevant to understanding hemoglobin function in a physiological context and are kind of at the interface between single protomer changes and changes in the contacts between protomers in the multimer. These are the famous Bora proteins. Those of you who are fans of quantum mechanics, the name Bohr will attract your attention. This is not Niels Bohr. This is his father, Christian Bohr, who made the fundamental early physiological discoveries on which our current understanding of the Bora protons and their role in regulating hemoglobin is based. So this is a modern reconstruction of one of the observations that Christian Bohr originally made. This is a oxygen saturation curve of hemoglobin. Notice it has the typical sigmoidal, not hyperbolic shape that we've come to expect. And what do you notice here? That as pH goes down, from in this case from 7.6 all the way down to 7.2, each decrease in pH, that is each increase in hydrogen ion, remember pH is the negative log of the hydrogen ion concentration. What happens? The curve shifts down. In other words, if you take a particular vertical line, say the dotted line that's shown there, each increase in hydrogen ion concentration causes an increased release of oxygen. That's going to turn out to be really important physiologically and also to understanding how the molecular details of designing a machine that can produce this nice sigmoidal response. So let's look more closely at that. So something remarkable happens in red blood cells, in the erythrocytes that carry hemoglobin from the lung to the tissue and back again. There's an enzyme called carbonic anhydrase. And what that enzyme does is catalyze a reaction that's freely reversible, but normally it occurs very slowly in the absence of catalysis. For physiological reasons that will emerge here in the next couple of minutes, we want it to occur rapidly in a functioning circulatory system. So what carbonic anhydrase does is cause rapid equilibration between CO2 and water on the one hand and the acidic bicarbonate ion on the other. So you take one CO2, one water, both neutral molecules, combine them to make bicarbonate, releasing a hydrogen ion. You've turned two neutral molecules into an acid. So when CO2 concentration is higher, as it is in actively respiring tissue, remember, and as we'll return to in much more detail later, one of the central processes in all of metabolism is taking oxygen, using it to burn reduced hydrocarbon food, turning that oxygen into CO2, among other things. So that's the fundamental process that the oxygen is feeding here.